Greetings from the Center for Excellence in Education. Thank you for joining what promises to be an exciting and informative Zoom session today about unraveling DNA, the world of genomics. I am Joanne DeGennaro, president and co-founder of the center, with the late Admiral H.G. Rickover. Our mission since our founding in 1983 has focused on STEM education to nurture and encourage high school and university students for leadership and excellence in science, technology, engineering, math, computer science, and business. I welcome you to review our website, newly designed at www.cee.org. Learn about the programs we just prefaced, RSI, USABO, our Teacher Enrichment Project, and our advisory and consultancy. There are so many ways for you to become involved with us, other than just donating funds to us, although we won't say don't do that. All our programs are cost-free to participants in our programs. The center is honored today to collaborate with the nationally and internationally recognized corporation Illumina for this webinar, and for Illumina's eminent researchers, scientists, administrators to participate with our alumni of USABO and RSI, who will address you today about the advances in genomic sequencing. I also thank CEE's program leaders for their hard work on this project. Dr. Michelle King, who is manager of the USABO program for nearly 10,000 US students who participate every year, and Kim Edwards, our manager of the Teacher Enrichment Program for rural and urban teachers that has already been in 12 states. So you've heard from me enough. Let's begin this very exciting program. And how can I thank the nearly 300 participants who will be with us today? Take it away, Michelle and Kim.
Thank you so much, Joanne, for that wonderful introduction. We are so happy to have all of you here today to celebrate World DNA Day with us. I just wanted to let you know, we will be taking questions from the audience, so please submit all questions through the Q&A function. And we are thrilled to partner with Illumina for their Future is Bright campaign, inspiring the future generations of genetics leaders. And it's my pleasure to introduce our first set of speakers, Dr. Ashley Van Zeeland, who is the Vice President of Product Integration and Customer Collaboration, and Dr. Cande Rogert, who is the Vice President of Scientific Research. And please join us and as they lead us through the evolution of sequencing technology.
Thank you so much, Michelle, and thank you so much to the Center for Education Excellence for the invitation to participate today, especially because today is DNA Day. And so when you work at Illumina and you've spent your career in genomics, there's almost no more exciting day than DNA Day. And so to kick us off, I thought I would share a short video just to help ground us all in the incredible scale at which our technology operates and how much information is encoded in every cell of our body. And so many of you probably know this, but as you know, in this video we zoom hopefully here into the bloodstream. This is a common way that we actually identify and extract DNA for profiling and sampling. So we go into the nucleus of the cell and you have your chromosomes very tightly packed. As you unwind those, you start to open up the very tightly packed chromatin structures. And it's not until you unwind that even farther that you get down to the base pairs of A's, T's, G's, and C's that encode the basic software of life. And so I really just think it is an incredible feat of technology and an innovation that we're now able to routinely get to that scale of resolution for an entire genome's worth, or 3.2 billion base pairs of information for a human whole genome, for about $600. It wasn't that long ago when that was almost unimaginable. And so this timeline slide shows you just a quick history of what that technology evolution has been like and the kinds of applications that it's opened up. And so if you look back, the very first piece of technology that you see there in 2004 is what's called a SNP array. And what that is a kind of technology that looked at individual mutations. And so this one looked at 1,536 individual mutations across the genome, across 96 samples. And that was completely revolutionary at the time and displaced the existing technology, which only genotyped 3 SNPs at a time. And for the record, it's that technology that I used in my PhD thesis work. And so I was blown away when I saw this hit the market. And it wasn't too much longer after that that the array went from 1,500 markers to 1 million or more. And then it got really exciting because then we could do things called genome-wide association studies and start understanding the genetic basis of some common diseases because this was inexpensive and it was genome-wide. And then a major turning point happened in 2006 when Illumina bought a company, Solexa, and Dr. Rogert can speak more to this in her background, but that really changed the game for churning from just looking at a handful of mutations across the genome to actually sequencing every base pair. And at that time, it would take about 3 days to produce 1 billion bases of DNA, and you want to sequence every base a couple of times. And so you could sequence a whole genome on an instrument like that. I think I calculated this might be around 9 months. Okay, which was, still pretty mind-boggling because the first time we ever sequenced a human genome, it took about 10 years and about $3 billion. So to have an instrument that you could put in a laboratory and actually turn out that amount of genetic sequence information directly was really a game changer. From there, you started to see new applications of sequencing technologies start to be applied. And it was applied to things like cancer, which is really a disease of the genome, to understand how mutations can drive cancer growth or how you can target specific mutations in tumors to selectively kill those cells. We also saw the first application in 2011 of sequencing applied to basically save this young boy's life. He's right there in the middle. Sweet little face, Nicholas Volker, had a rare and undiagnosed disease. And genome sequencing was able to identify the causative mutation, identify intervention, and save his life. And so a lot of things started to unlock very quickly as companies like Illumina and others continue to really drive the cost of that sequencing down. And so as I mentioned, today we've released a number of instruments. So if you look at 2017, we have the big box, the NovaSeq. Last year, we released that beautiful looking box there above 2020. That is the NextSeq 2000. And each of these can turn out just a huge amount of genomic information. The NovaSeq, again, a whole genome for about $600, which when I started was just, like I said, completely mind-blowing. So that's a little bit about the technology, but I want to talk about the future. Okay, so the past is wonderful, but if you can go to the next slide, what's really exciting is if we think about where we can go. And this is a big reason why when I had the opportunity to join a company like Illumina, I jumped at it. So my background is in neuroscience and neurogenetics. As I mentioned, I was doing my PhD research, kind of looking at one mutation at a time, but really saw the opportunity to probe more deeply into developmental neurobiology by looking at the full sequence of information. And so that's where I started to get really excited. And that's where you start to run into the intersection of genome science, and computational science and life science. And if you just look in the middle column here, we have the cost per genome. So as that cost falls, it becomes increasingly clear that we need more data capabilities to analyze this to meet the pace of the technology that we're releasing. And so I had the opportunity to actually start a company in the genome analysis and interpretation space after my postdoc, built that company up. It was acquired. I managed the build out of a platform for a large sequencing effort, and then joined Illumina. And so when I look at some of the things we've talked about for our future sequencing roadmaps, where we've talked about we can increase the density of our core flow cell technology. And what that allows us to do is output more data for the same unit of area. We can also increase our accuracy. So I talked about applications in clinical spaces like cancer or rare disease diagnosis. We have to maintain incredibly high accuracy with high sequencing output so that we can really use this to unlock clinical applications and be confident in diagnoses or confident about the genetic information that might target a therapy that a doctor can prescribe. We can also look at chemistries that allow us to go faster, or information that allows us to read through the harder, trickier parts of the genome through things called long reads. And so we're still at the very, very beginning of this march. And so you can see we have a little bit of a dotted line there in the middle column to contemplating something like $100 genome someday. So today it's under 1,000. If we unlock $100 whole human genome, starting from where it was at $3 billion, I mean, that's a massive technology innovation. It's an honor for me to get to work with the teams of scientists and engineers and so many others at Illumina who make this possible. And then I think about what the next phase of $100 genome world will look like. What kinds of applications can we help our customers to pursue? And so I think we're going to hear more about that today. And now I'll hand it over to Dr. Rogert.
Thank you, Ashley. And it's my pleasure today to be with you guys to talk a little bit about DNA and the history and how innovation comes about. So innovation is driven by diversity. And when I was starting my career, I graduated from my PhD from Manchester University in the UK. I knew I wanted to be in technology, and I didn't quite know how a company like Illumina or any tech company in biotech creates technology. And really, what I learned through the years is that innovation comes at the interface from different core sciences. And I think that that is what Illumina does extremely well. Our teams are greatly diverse, and we encourage each of those functional teams to work really closely together. And like Ashley told us, in that latest platform that we launched, the NextSix 2000, for example, it was an incredible feat done by combining the efforts of biochemistry and fluidic design and nanofabrication and optics and software and analysis. And out of that, what came out is this ability to innovate and invent. And when we look at that and we try to see how diverse our scientific population in Illumina and our scientific achievements are, we saw that for all of the inventors, we have around 327 inventors out of 2020 at the end of last year, and they're scattered across the globe. So we're pulling from every different location, diversity and know-how, and trying to bring all of that together. When we look at how innovation is done, there isn't like one person that invents everything. It's just groups of people working together. Roughly, there is 130 employees that are named inventors and patent applications per year. So, and many of those patents have many names in them. There is very, very few that have only one or two. And when we look at the organization that I work in, which is research and technology development, 46% of our employees are inventors. So we're constantly catalyzing new innovation. And something that makes me really proud, we're still not like perfect, but our number of Illumina female inventors is twice as high as that of the national rate. for women receiving patents in the US. So there's still work to do to have a 50-50 split between females and male inventors at Illumina, but by continuing pushing, continuing encouraging new ideas across multiple technical fields, we know that we're going to be able to come up with better and better technologies to serve the genomics fields. If we move to the next slide. And like Ashley said, as the genomic history evolved, more and more data was generated. And when we think about the genome is 3.2 billion bases, but in order for us to read it, we have today our sequencers. And as we keep reading it and becoming more versed in it, on this, reading this book, we realize that there is much more richness in the information that we're getting, that we want to continue digging deeper and deeper. If we click twice, I think there is some animation in the slides. Thank you, or just once, there. Thank you. So as the data continues to increase, the amount of data continues to increase, what we realize is that we are enabling new fields and we're enabling new disciplines that before were not typical of genomics or biology itself. But we needed to start working more deeply in fields like the field that Ashley comes from, on data science and analysis. So more data comes out, the scientists and the biologists want more completeness of that data, more diversity on the data sets that they're getting, such that they can make more complete assessments of those genomic insights. And at the same time, because we're generating more data, analyzing of that data becomes more complex. And that gives rise to complete new fields in genomics. So combining data science with genomics, what we started doing it over the last few years, is very recent, is trying to understand the importance of that variance. So some of you may know our genome is 3.2 billion bases. We have 2 copies of it, so it's 6.4. And all of us have small differences in our genomes. Those are called SNPs, or if they're larger, they're variants. And because we all have many variants, we know that most of those variants are benign. They do nothing. They just are differences between different people. But there are a few that we don't know what they do. And what we're trying to figure out is, like, are those variants cause of disease or not? So in that particular topic, a new field of data science and trying to understand significance of variants evolved over the last few years. And scientists are working really hard and really intensely in trying to understand which of those ones we should pay more attention to. But in order to do that, that data science and big data field also exploded. And you can see it on the graph on the right over there. We had to help our customers, who are biologists and doctors and geneticists, trying to manage that data, organize it and analyze it in a way that they can not just look at bases, but actually work with insights and generate knowledge that overall will give us some wisdom over genomics. We can move to the next slide. And I think that last statement, generating wisdom in genomics, is also exploding new fields of biology. So before, we could only see a few SNPs here and there. Now we can read whole genomes, but we can not only read whole genomes. Now what we're trying to understand is that cells inside a body, depending on where they are, they may actually be expressing different genes in different manners. So new fields started coming out over the last few years, for example, in spatial transcriptomics, which is looking at tissues and trying to understand what is the genetic expression profile of those tissues, of the cells within those tissues, and how different cells talk to each other. So with the the advent of genomics, being able to reduce the cost of a genome from $3 billion to $600, not only made whole genome sequencing more amenable and actually an aspect of biology and genetics that people are very interested in, but it also created new fields, like, for example, single cell analysis, spatial transcriptomics, trying to understand the relationship between position and expression and disease or function. And at the same time, the ability to do all of this, put all these technologies together, started creating completely new fields, like enabling synthetic biology. Now, when a scientist makes a change and creates a new gene, they can actually read it, understand how it works, and understand the impact that it has on the system that they created. And the same thing with CRISPR and genome editing. Now it's actually possible to modify a particular gene, changes sequence and function, and in that way, actually, hopefully in the future, prevent diseases that up to now have not been possible to be prevented. So large-scale genomic sequencing has enabled this possibility of understanding how the environment, how the genomics itself, how the epigenomics, which is the modification of the DNA inside our cells, how transcriptomics, which is the expression of certain genes in different cells, proteomics, which is the proteins that we actually have in our body, and metabolomics, in conjunction with the environment, alter the phenotype of a particular cell, organism, or tissue. And that is creating that revolution in biology, where soon, if not now, we're going to be able to actually associate a particular sequence or expression pattern with a particular phenotype and function in the body. And 10 years ago, that was considered science fiction, but I think we're very, very close to that. And I thank you for listening to us talk today.
Awesome. Thank you so much, Dr. Van Zeeland and Dr. Rogert. It's so exciting to hear about the technological advances, and I know I'm very excited to see what our future holds. And so our next set of speakers, I'm incredibly jazzed about. They are a set of incredible researchers who are leading the way in genetics research and how it relates to our human health. So first, I would like to introduce Dr. Mimi Shirasu-Hiza, who is an associate professor in the Department of Genetics and Development at Columbia University, and she attended our Research Science Institute in '88. Dr. Jonathan Gootenberg, who is a McGovern Institute Fellow at MIT, who attended the USABO Olympiad in '08 and '09, and his incredible co-PI, Dr. Omar Abudayyeh, who is also a McGovern Institute Fellow at MIT. And up first, we have Dr. Mimi Shirasu-Hiza, who will speak about the circadian clock behaviors and how she investigates it using the model organism Drosophila melanogaster.
Thank you so much for that kind introduction, Michelle. Yes, I was an alum of, or I am an alum of RSI. I have to say RSI was a turning point in my life. It really was. Way back in high school, we didn't really have to say the date, but that's okay. Because of RSI, I majored in biophysics and biochemistry instead of comp lit. That's a true story. All right, so I'm gonna try and share my screen. Things could have turned out really differently for me if I had not done RSI. That is the truth. Oops. Oh. Okay. Let me know if you can't see this. And turn this to, there we go. All right. So my lab works on circadian regulated physiologies and their contributions to disease in the model system of the fruit fly. as Michelle said. I'm just going to tell you about one project that's going on in the lab. This is our sleep project. The reason I'm going to tell you this is it's really appropriate for today, because we got to a certain point in the project where we realized that the only way we were going to solve the problem was to use functional genomics. And in fact, as it turns out, the Columbia Core, where we do all of our sequencing, uses Illumina technology. So it's the best. I'm going to have to warn you, we don't have the answer yet. So I'll take you up to the point where we are now. It's a work in progress, but hopefully we'll get the answer soon. All right. Okay. So the question that we started with was, what is the function of sleep, right? Why do we sleep? We spend maybe a third of our lives doing absolutely nothing. And if you look at this picture, all of these animals have been shown to sleep, and more, many, many more, right? Almost all animals sleep. So through evolutionary time, sleep appears to be a really conserved behavior. That's strange, because sleep doesn't do anything, apparently, for fitness, right? Why would it be selected for? Why wouldn't it be selected against? Why wouldn't we eventually evolve so that we didn't sleep all the time and spend 1/3 of our day not running from predators, not looking for mates, not making babies, not taking care of our young? All of these things that are important for the survival of the species are not being done for hours and hours every day consistently, right? Why would that behavior of sleeping still exist today, right? So that seems like a mystery. That seems clear. Sleep has a function, we just don't know what it is. And that function is what's been selected for throughout evolutionary time. The other clue that we have is that defects in sleep and circadian regulation are associated with disease in humans, right? I mean, you and I know if we don't sleep, we feel like terrible. So intuitively, we feel like sleep has a function and sleep is important. If you look at many different diseases, for example, neurological diseases, Parkinson's, Alzheimer's, Huntington's, schizophrenia, Alzheimer, epilepsy, bipolar disease, autism. If you look at metabolic diseases, diabetes and different cardiovascular diseases, and even cancer, right? There are associations with defects in sleep and circadian regulation. So intuitively, we know that sleep is important, and epidemiologically, we know that sleep is important. At the same time, we don't have a specific physiological function for sleep that we can attribute to with a molecular mechanism. So we set out to investigate this using Drosophila, and here's a picture of a dime. I took this picture myself because I thought it'd give you a good sense of scale. And this is a Drosophila. This is a male Drosophila. Actually, we use mostly males in our studies just for technical ease because they don't lay eggs, which turns up the food. All right, the reasons that we think Drosophila is a great model organism to study sleep in particular, fly sleep is actually really similar to mammalian sleep. And I always get the question, people are always like, what, flies sleep? Yes, flies do actually sleep, and they sleep in a way that is similar to us. So they sleep at night and they walk around during the day, and they have breakfast and dinner. They don't usually have lunch, but they have breakfast and dinner. So they're very regular in their circadian rhythms, which are sort of behaviors that oscillate with a 24-hour cycle. And they are also pretty regular about getting their sleep, right? There are also very powerful genetic tools that have been established in the fruit fly for 100 years, which is great for people who want to manipulate gene expression like us. And then finally, in the last 5 or 10 years, a number of sleep mutants have been made publicly available and have been identified through forward genetic screens. Not by us, we haven't discovered any of these genes, but these are tools that we can then use to study the function of sleep. Right? And these are some of the references here for people who have discovered sleep mutants. All right, so this is a list of sleep mutants, and I don't want you to read this, don't read this. It's written in very light colors because I don't want you to read it. I just want you to get a sense of the diversity of sleep mutants that are out there, right? So we picked up a number of these from our collaborators and colleagues. They work in different pathways, right? In the brain, mostly, they affect neuronal function in different ways, but they're all different is the point, right? And the reason that we wanted different types of mutants is because we figured if we look for a common physiological defect amongst these very diverse set of sleep mutants, we might be able to find the common physiology that is tied to sleep, because the common defect that they all have right now is that they're short sleeping. So all of these sleep mutants are short sleeping. They retain mainly their circadian rhythms, but so they sleep on time, right? They sleep at night. They just sleep in really either discontinuous bouts or not very, very long, okay? So short sleeping, very different. If we stress them out in different ways, all of them, if there's a function of sleep that is being required to deal with the stress that we are giving them, then they should all be sensitive to that stress. Does that make sense? That was our sort of naive hypothesis going in. And we tried a number of different stresses. My grad student was a little bit tearing out her hair for a while. And it was kind of a hot mess for a long time. There was no consistent defect until she tried oxidative stress. So I'm going to walk you through this. This is a graph. It's a simple survival curve. On the y-axis, you have percent survival, and on the x-axis, you have time in days, okay? And I'll show a few of these, and the controls are always going to be in gray. So those are just wild type, right? Those are just like typical non-short sleeping, regular sleeping flies that are genetically related to the mutants. Mutants are always going to be in a color. So this is insomniac. The names for Drosophila mutants are really great. Insomniac is a short sleeping mutant. And as you can see, this curve, the pink curve, is shifted down and to the left, indicating that it is dying faster than the controls, right? And there, for people who are interested, the p-values are here. So what we did was we applied a toxin, a pesticide actually called paraquat, that is associated with Parkinson's. We applied this poison to the flies and watched them, counted their death, sorry about that, and watched for their survival and looked to see if there was a difference in how they survived. And consistently, the insomniac mutants died more quickly than the controls. Oxidative stress, just as a note here, is a perfectly normal process that does happen in your bodies due to metabolism when you have reactive oxygen species generated, right? We all try to eat lots of antioxidants so that we can decrease the damage that these reactive oxygen species might do to other macromolecules in the cell, right? But in fact, the cell has its own defenses that are way more powerful than any antioxidants that you could eat. Those antioxidants are what generally defend the body against reactive oxygen species. So oxidative stress is when those reactive oxygen species are too much for the cell's defenses and cause a lot of damage and leads to, in general, death. And this is what happens with our fruit flies. So we said, well, this is a pretty dramatic phenotype for one mutant. What happens with all the other mutants? It turns out, again, you don't have to look at every single graph, but the trend here you can notice is that all of the colored lines are significantly lower and to the left, shifted to the left, relative to the gray lines. And that told us that all of our short sleeping mutants are sensitive to oxidative stress. In other words, we found a common defect that is common for all of the short sleeping mutants. And to this day, every single mutant that we have tested that is short sleeping, whether or not it's a known short sleep mutant or not, is sensitive to oxidative stress. And we tried many different kinds of oxidative stress, and it was true for every type of oxidative stress that we tried. So this is a really incredibly consistent and dramatic phenotype. It's not causal, right? Yet. So this is the question that we had to ask. Does it go the other way? You can imagine that, for example, some defect might cause both sensitivities, right? And in fact, you could have sensitivity to oxidative stress might actually cause short sleep, right? So the question that my graduate student asked then was, can we increase sleep? And if we do that, does that increase survival of oxidative stress. That would suggest that in fact, there is some mechanism that is occurring during sleep that is increasing or enhancing the body's defenses against oxidative stress. All right, and so we did this in two ways. The first way was genetic. We stimulated sleep regulatory neurons, sort of made the flies sleep a little bit more, or we gave them a pharmacological agent, gaboxidol, that is a GABA-A agonist, that increases sleep. Just sort of made them have a little longer nap than usual, right? Then we tested their response to oxidative stress. And you can see here on the left, again, the colored lines are going to be the experimental, and then the gray lines are the controls. You can see that the stimulation of sleep or enhancement of sleep led to the colored curve being shifted to the right and up, showing that these flies are now resistant or more better able to survive the oxidative stress. Okay, does that make sense? Right, so using these two methods, we've now shown that sleep is important somehow for defense against oxidative stress, but how? That's the question, right? What is the mechanism underlying it? And we do not know, but we realized after trying many targeted experiments, trying a candidate approach, and just as our two previous speakers were saying, there are a lot of possible candidates. We decided that we had to go with the whole genome and do an unbiased look at what was happening in terms of gene expression. So I'm just going to explain that in a second. So we're calling this functional genomics. Functional genomics looks at how DNA changes and is being expressed in order to understand disease, or at least in our case, to understand disease. Specifically, we're looking at RNA sequencing, okay? So most of you probably know this is central dogma, but DNA is transcribed into RNA, and RNA is then translated into protein, right? And in different tissues at different times in the organism's life, different genes might be expressed a lot or a little or not at all, or, it might change, right? And so this is the thing about circadian regulation, which is, the wheelhouse of my lab. Gene expression changes in your body, in every tissue in your body, actually, over time, over the circadian day, right? And so your skin has a clock, your heart has a clock, your hair follicles have a clock, your brain has a clock for sure. And by clock, I mean a mechanism that figures out what time of day it is and changes the expression of your DNA. So your DNA is static, your DNA is exactly the same everywhere in your body, but how it's expressed, how it manifests is going to be really different in different parts of your body and at different times of day. So here I'm thinking about like a single gene, right? And it might be expressed more at night than during the day. So you have this oscillatory pattern over 24 hours, right? And so here's DNA being transcribed into RNA, which is are the messengers for gene expression. It might be different if you check 3 or 4 hours later. And then it might be different through the entire circadian cycle, right? The thing is, the gene expression over the circadian cycle is going to be different between our wild type and our mutants, not only under normal conditions, but then also under oxidative stress conditions. And by doing the comparison between all 4 of these groups, we can hopefully, and we're doing this actively right now for 2 different sleep mutants and their wild type controls, we're going to be able to see exactly what the molecular changes are that might underlie the defects in defense against oxidative stress that we're seeing a physical manifestation of in their survival time. Okay, so I just want to wrap up there and acknowledge the people who did this work and my partner in crime, my very close collaborator, Julie Camden. And these are the people in our lab. We sort of have a joint lab as well. And this is Reed O'Connor and shoot, it's not, I can't see it on my screen. Here we go. Vanessa. Vanessa was the lead graduate student on this project, and she really was an awesome person to take the lead. And these are other authors. All right, thank you.
Awesome. Thank you so much, Dr. Shirasu-Hiza. It was so interesting to learn about the circadian rhythm, and I'm glad that I can say that I need more sleep to better my health. And without further ado, I want to introduce our next set of speakers, Dr. Jonathan Gootenberg and Dr. Omar Abudayyeh, who will be talking about developing new tools for treating genetic diseases. Take it away, guys.
Great. So let me just share my screen here. And
great, yeah. Thank you for having us today. The kind introduction.
Is that presenting? All right. Great. Well, thanks for that great introduction. And we're happy to talk today about kind of one of the other sides of the DNA story. So of course, it can be very useful for discovering really amazing biological phenomena, such as the circadian rhythm, but it can also be very useful for discovering and developing biological tools. So that's what we'll be chatting a bit about today. So as I'm sure you appreciate now, there are many, many genomes that have been sequenced. And this is a human genome, of course. This is a karyotype, but you know, humans aren't the only organism, of course. So it's really something that is becoming very valuable as a resource, both for understanding the biology of different diseases, but also potentially for treating them. So as we understand more about this biology, we understand that, of course, all of us as humans are somewhat different. So there are genetic differences, and many of those can underlie disease. So often a single base pair change in your DNA can lead to something like sickle cell disease or cystic fibrosis, or there are a multitude of potential eye degenerative diseases. And these can be due to as little as a single base pair change. In the case of sickle cell, you know, it's one amino acid change. Of course, that's not the only thing that can happen. You can have larger pieces of your genome be duplicated or deleted, leading to different diseases. But it's important to keep note of what these potential different mutations can do, but also that we could potentially correct them. So that's on one end, but of course, there are also genetic differences that are actually valuable in a biological sense. So here, there are genetic diseases or genetic differences that lead to protection. And some of these are a little bit more simple in the terms of what they are in the genome. For example, HIV, there's a single mutation, it's actually a deletion that leads to resistance in the CCR5. It's the CCR5 delta 32 mutation. But there are also much more complex different changes that can be involved. And GWAS was mentioned in our previous panelists. The idea that we can start to collect such a large amount of information about the genetic differences in people and be able to map those over to different developments or lack of developments of diseases allows us to really discover other modulators of disease. For example, in Alzheimer's disease, there are protective mutations in ApoE that lead to a reduced function. And in diabetes as well, there are mutations in certain genes that lead to reduced occurrence of diabetes or reduced severity. So this one comes from both a really great mechanistic understanding, but just the idea that we get so much DNA out there that's sequenced and that we can start to really see what is happening. And of course, this isn't only applicable to human health. There are many different traits in agriculture, for example, that allow us to actually design and engineer traits. Now, selective breeding in agriculture and livestock certainly predates the development of DNA sequencing, but now this gives us the capability to understand what these traits that we're actually affecting, what is the underlying mechanism of them, and to actually rationally design new amazing capabilities. One interesting aspect is even the concept of bringing back extinct or basically non-cultivatable fruits, so we can develop entirely new classes of interesting fruits or vegetables. So it's obviously a very interesting area. So, but all of this information really starts to paint the picture, really the demand for a tool. So your genome is 3 billion letters long. It's, well, 6 billion because you have 2 copies. And some of these plant genomes are even many more billion letters than that. So really we need the capability to not only understand the sequence of DNA using DNA sequencing, but also to selectively go to a place in that large genome and target it, change it, or modulate it. What we really need is a search bar. So if you're in Word and you want to make a change to a manuscript or a document or whatever you're working on, you want to go to a certain place, you can search for a word, a sentence, or even a set of letters and go there, and then you can edit it. You also might be able to copy it and put it somewhere else. That's really valuable because without that capability, we wouldn't be able to change that information. And if there was a typo in that document, we wouldn't be able to correct that typo, which could lead to something bad happen, like you failing an exam. So having this capability is very important. But how do we actually make this capability? And so that goes to the other end of the power of DNA sequencing, which isn't only to understand human or plant biology so we can make better crops or understand disease. It's really looking at this enormous diversity out there. So there's just this immense diversity in the kingdoms of life. And that allows us to really both understand these really intricate processes that lead to biology, but also to potentially take those processes and put them into different contexts. So this is just a tree of life from 2017, so it's actually outdated now. It's always evolving, but you can see that, you know, eukaryotes, the actual domain that we're in is only right there, and we're actually only a tiny part of that. So it's nice to think a little bit broader than just eukaryotic cells. So what does this really mean for biotechnology for developing tools? Well, by being able to understand the processes here, biotechnologists have actually been able to generate a whole suite of tools for different applications. So if you look at any of these different tools that are used in biology, the capacity to splice genes together to create recombinant proteins or do recombinant DNA, those come from natural organisms such as bacteria and phages. So developing restriction enzymes, polymerases, ligases, and other enzymes that are now the backbone of molecular biology, those came from nature. More recently, fluorescent proteins, which allow us to track different cells or proteins or other biomolecules inside of their natural contexts and to image them and see what happens, those come from natural organisms such as jellyfish or certain fluorescent bacteria. Of course, understanding energy and different applications in crops or biofuels is very important, and knowing exactly how these pathways work to synthesize different potential energy applicable molecules is very vital for the emerging approach to things like climate change. Then thinking about human health, how we can actually take and use things like viruses or other natural ways to get inside of the body to actually put different things into the body can be very useful for therapies to deliver a broken gene, or viral vectors are even used in certain coronavirus vaccines. The capacity for optogenetics to be able to control neurons or other electrochemically sensitive cells with light has been incredibly important for the development of many neuroscience applications. And then, of course, what we'll be talking a little bit about more today is gene editing. How can we use natural bacterial systems and other systems to develop tools to edit the genome, to understand and detect nucleic acids, and really to be able to develop that search bar that is so desperately needed to unlock a lot of new applications in human health. So I'll pause there and I'll turn it over to Omar to keep going.
Great. I guess, are you good to control the slides, or?
Yeah, I can do that.
Okay, great, thanks. So I guess, yeah, just diving into the next slide then. So as Jonathan was saying, you know, CRISPR is one of these really amazing tools that come from nature, but you know, the history of where CRISPR come from is actually really interesting. So you know, at its core, CRISPR can be characterized really by just a DNA signature in bacterial organisms. And that's this structure shown here of repetitive DNA, which is shown by the gray boxes repeating, interspaced by sort of unique sequences. And because of this, you know, unique DNA signature is given this long name, clustered regularly interspaced short palindromic repeats, or CRISPR for short, which is actually quite catchy, and the reason it's stuck. But what happened is in the late 80s, there was actually a Japanese research group that was starting to sequence some bacteria and trying to study the alkaline phosphatase genes in those bacteria. Nearby those genes, they actually saw this DNA signature. And at the time, that was the first time something like this was seen. And so it didn't really make sense to them. And I think they just noted it in their paper. But then in the 90s, as the sequencing sort of race was kicking off, and it's really relevant that Illumina scientists were on this call earlier, but before we had Illumina sequencing and next generation sequencing, we had much harder sequencing approaches, but people were still trying to sequence bacteria. And what they noticed is as they sequenced bacteria and sequenced the viruses that infect bacteria or phages, they started seeing these repetitive sequences actually correspond between the bacteria and the phages, or let me say the sequences between the repeats, the unique sequences. And so these connections between bacteria and viruses started to allow scientists to realize in the early 2000s that perhaps these systems have something to do with immunity or something to do with the arms race between viruses and bacteria. And so with that hypothesis that's involved somehow in bacterial defense, it took another decade or so to show that these genes can actually these sequences and genes can actually defend against viruses, cut the DNA or RNA of viruses, and that eventually led to the realization that if we have tools to cut and manipulate nucleic acid, maybe that can be used as a tool to better benefit humans. And so at its core, what's also really cool about this system is basically when the bacteria is infected by viruses, little pieces of the virus get chopped up and those pieces get integrated as these colored diamonds here. And so the bacteria is actually able to remember past infections and use those memories in essence to defend against future ones, because these colored boxes can actually be expressed as short RNAs that then complex with nucleases that can cut basically future infections. And so it's actually an adaptive immune response that's really useful, and it's that adaptiveness that makes it programmable when we use it for gene editing or gene cutting. So I guess jumping to the next slide. What's really interesting is sort of in, now we're talking about 2012, 2013, 2014, people started really honing in on Cas9 as this nuclease that can be programmed by these colored diamonds to go against, viruses, and people started trying to use it to cut human DNA and edit human DNA. But what's actually really interesting is, the CRISPR world is quite diverse. So although most people talk about Cas9, there's actually dozens of other types of enzymes and CRISPR enzymes. This is actually where, Jonathan and I have spent a lot of our time is trying to understand the diversity of these systems and how they work and what they do, and can we find new tools beyond Cas9 that could be useful for gene editing or RNA editing even. So, and I think what the most interesting thing is most of the systems up until 2012 have been the complicated ones where these class 1s. That are all these red proteins, and then the bottom are the class 2s, these much simpler ones. And there weren't too many of these simpler versions known, which are the ones that are easier to use as tools. And a lot of our work has been trying to expand that understanding. So I guess going to the next slide. So basically by being able to uncover new CRISPR enzymes, like, there's Cas9, we've uncovered now Cas12 and Cas13, we've been able to make quite a toolbox of different types of technologies. So I think, there's a lot covered on this slide, but generally like kind of on the left, you have Cas9, Cas12, Cas13, which can cut DNA or RNA. And then you can start adapting those technologies to do other things. So that if you move to the right, there's this CRISPR A and CRISPR I. You can actually, instead of using CRISPR enzymes to cut, you can use them to bind DNA and then recruit transcriptional activation domains to turn on genes, or recruit, repression domains like, CRAB, or epigenetic modifiers like methyltransferases to actually repress expression. So now we can talk about using Cas enzymes not only to cut and knock out things, but to actually change the epigenome and change the structure of the genome and change the marks that are on the genome, which is really exciting. People have also going now up towards the right side, more on the right side, base editing. So the base editing on the right, so DNA base editing, you can actually use these catalytically dead enzymes to now recruit deaminases So there's all these naturally occurring enzymes that can actually change the atomic structure of nucleotides, like, you know, the adenosine, the A's, the G's, the C's, and T's. And by recruiting those enzymes to the genome, you can now convert, you know, an A to a G, or, you know, a C to a T. And so if there are mutations that someone has, you can actually do very precise atomic level base changes, which is really cool. And not at all what these enzymes were naturally evolved to do. Repair and rescue listed here are actually using Cas13. So Cas13 is an RNA targeting enzyme, and there you can recruit deaminases to target RNA. And the vision for RNA is, you know, RNA is not permanent. And so if you could do gene editing in RNA, it's a more temporal thing. So you don't have to worry about permanent off targets or permanent modifications that we don't fully understand. And so one day, I think the vision there is if we could have a pill for gene therapy. So you take that pill and you get your base change, or, you know, your mismatch correction, but you can always stop taking that pill if for some reason we realize that we don't want to have that mutation any longer. And so then moving now towards the middle, there's technologies for CRISPR diagnostics. So Sherlock is one of those versions, but a lot of these CRISPR enzymes, people have only thought of them to cut DNA and edit DNA, but they're very good at, you know, recognizing very rare sequences in the genome that's, you know, billions of bases. They're actually a really good search function. And so instead of saying like, hey, we want to find a sequence and edit it, what if we just say, hey, we just want to find the sequence and tell us that sequence is there? And so a lot of people have started, you know, including ourselves, have pioneered the use of CRISPR for diagnostics. And I'll actually talk a little bit about our COVID diagnostic work with using CRISPR. And then in the middle, this is kind of now the frontier, this Cas system, but there's actually people starting to realize there's Cas proteins that interact with transposases. So they're a programmable transposition, which is basically gene insertion. So mobile genetic elements are these elements that can basically jump around genomes, and they're not quite like viruses, but they can, you know, replicate and insert themselves where they're not supposed to be. And so now the idea is if we could have programmable gene insertion, you could start, you know, for therapeutics, actually if someone has a mutated gene, you could replace that gene copy using these Cas associated transposases. That's kind of like a very recent finding that people are working on. I guess going to the next slide. So I talked a little bit about, you know, using CRISPR diagnostics. Our technology is called Sherlock, and we can really use it for all sorts of very sensitive and specific, you know, infectious disease detection, like viruses and bacteria. We've been trying to make them point of care. So, you know, the vision one day is that everyone can have a little testing device at home where you can test yourself. You know, if you have a cold, you know, you might worry that's pneumonia, and so you could, you know, tell yourself, you know, or test yourself and figure out if you have pneumonia or just a cold or strep throat. And also, of course, you can use this for cancer detection or genotyping or whatnot. And I think, you know, what's cool about CRISPR-based diagnostics is they're really cheap, and we've been able to adapt them for even like paper test strips as well. So they're quite a flexible and easy to use technology that we're excited to hopefully keep developing. Going to the next slide. We actually, when the pandemic was starting, January 2020, we started working on a COVID version with Feng Zhang at the Broad Institute to develop a CRISPR-based test for it. And we actually did some innovations around a CRISPR enzyme that comes from bacteria found in rotten fruit juice. We actually wanted a CRISPR enzyme that survived at hotter temperatures, so you can mix it with amplification reagents that operate like at 60 degrees. And so we're able to make this really simple master mix that you have to run at 60 degrees. And all you have to do is basically swab yourself, mix the swab with an extraction buffer, beads in the extraction buffer, bind nucleic acid released from the virus, which is step 3 here. And then we can resuspend these magnetic beads in our CRISPR detection master mix, which if you run for 30 minutes by fluorescence, you can get a result, or it's more towards an hour for a paper strip. And I think we tested close to 400 patient samples and showed a sensitivity, I think around like 93% on a very wide distribution of viral loads. And so I think this shows the power, you know, with biotechnology getting more and more programmable, right? The idea of programmable therapeutics, programmable diagnostics, we can really, as simply just, once you have the sequence of a virus, you can just go design an assay in a week. You have all the reagents to test your assay, and in maybe a matter of weeks, you have a new diagnostic kit, right? Or you have a mRNA vaccine produced. So I think the power of these types of CRISPR enzymes and other types of programmable systems is the ease that they can be used to rapidly be deployed for not, you know, new threats or things that come up. So I think this is kind of a nice demonstration of that. I guess going to the next slide. So in general, our group, Jonathan and I, are really focused on building out DNA and RNA targeting tools. The upper side is supposed to be the RNA toolbox, which we're trying to fill in more since the RNA-based CRISPR tools are more nascent. But I think this gives you an idea of, what we're thinking about as we try to build out these sort of biomolecular technologies. And then on that, just, I think a note about, we also are really just excited about how much unknown there is in the bacterial world and just, the tree of life here. We have a lot of computational projects where we try to mine, hundreds of thousands of organisms to find interesting new gene functions based on domains and whatnot. And I think there's still so much more to do. So, I guess for the, younger generation here, I think this is the idea of bioprospecting and just like searching all these unknown genes that are out there is really exciting for biotechnology and medicine. So, and then with that, I just want to say thanks to our entire lab. Some of the members are listed here, and if there's any questions, we're happy to answer them. So.
Well, thank you so much, Jonathan and Omar. That was so exciting to hear about how you're using DNA and tools to advance our health and medicine. So now we are moving to the Q&A portion of our webinar. So if you have any questions for any of our speakers, please submit them to the Q&A box at the bottom of your screen. I know a few of those have already been fielded, so hopefully we'll be able to answer your question. So up first, we have a question for Dr. Shirasu-Hiza. So the question comes in dealing with your model organisms. So what are the differences between studying circadian rhythms in a fruit fly and then applying it to a human?
Oh, yes. So I just answered a question similar to this in the Q&A, but I think of our flies as little humans, but I know that they're not. They're significantly different from humans, right? So there are some aspects of disease that we just can't model. For example, flies don't have an adaptive immune system. They only have an innate immune system. So they don't have T cells and B cells. They don't have dendritic cells. They don't have antibodies, for example. So we can only study the effects of whatever gene manipulation we do or whatever, physiological manipulation we do. We can only examine the effects on things affected by the innate immune system, for example. I think for us, it's mainly the technical efficiency of the tool. And that allows us to do so many things so quickly. But we do have to be very, very specific about how we apply our findings. I usually say in fruit flies, for example, don't take this home and start doing it to yourself, for example.
Oh, thank you so much. I know I worked with C. elegans, and that was also interesting in working with the model organism. So our next question
is that if we think about evolutionary conservation, anything that we find in the fly is going to be important for humans. We might miss things in the fly that are important in humans, but the things that we do find that are conserved between flies and humans are going to be important, if that makes sense.
Yes, thank you so much. So our next question is for Dr. Gootenberg and Dr. Abudayyeh. So for your vision for the futures of your technology, so would it be possible for us to detect genetic disorders like autism, ADHD, cystic fibrosis in the womb and then treat them in the future?
Yeah. I mean, I think so. I think that's actually a great question. It's something that does come up, especially in like ethics conversations around gene editing. But I mean, there is a big question of, why do you want to edit someone when, you could just do IVF screening one day and find the variants before someone's ever born. But I think, the reality is I don't think the entire globe is going to switch to IVF screening anytime soon, right? So although I think that's like a very, futuristic thing where you could do this outright from the beginning, I think somatic gene editing is definitely definitely going to be a big need for the foreseeable future. And that's obviously why a lot of people are working on it. I think a related question to this is, what is the therapeutic window for a lot of diseases? Because some diseases, like, when someone's an adult or even, late adolescence, it might be too late. Even if you go in and gene edit, the damage is already done to the cells. And so that's something you have to think about is, is it better to edit in the embryo or in early childhood, or that, that's very, very disease specific. But
awesome. Thank you. So I am excited about the world that this technology will open up. So our next question is for Dr. Cande Rogert from Illumina. So can you speak to the STEM fields that are involved in developing new technologies? Is it just biologists and microbiologists, or are there others?
Oh gosh, no, there's lots of fields. So I can give you a brief example of my team. So my team does new material science, and that is very broad. So the fields that we would be recruiting for our team are chemists, and that could be from synthetic chemists to physical chemists, biochemist, biophysicist, physicist, data science, structural biology, and protein engineering. It's just endless. And engineering, like fluidic engineering, mechanical engineering, electrical engineering. When you look at and I should have put together a slide of the actual scientific makeup of one of our platform teams, you probably have every degree that is in STEM being part of developing some of these platforms. And I think that is the beauty of technology development in biotech, particularly in this industry, is that you can tap into all these fields and have people that have their support system, basically their day-to-day management within their field, but they do most of their work across fields.
That's so excellent and so inspirational. It's great to know the team behind, a single piece of technology. And the next question we have, again, brings it back to Dr. Gootenberg and Dr. Abudayyeh. How accurate is CRISPR at detecting and replacing DNA? Are there problematic sequences, or do you foresee that there could be some complications in the future due to what we may learn?
That's a really good question. I mean, that's an immense consideration when you're editing DNA. Well, I guess one aspect is when you talk about replacing DNA with CRISPR or editing DNA with CRISPR, you're really leveraging your body's own DNA repair machinery for many of those edits. So sometimes that process can be a little leaky and have unpredicted edits. But there's another consideration, which is that the enzyme can go to a different place in the genome. If you're trying to target one place in the genome and you have another place that's very similar, the enzyme could go and actually edit in that area. And that's certainly a very important consideration, because if there were to be a permanent edit made to someone's DNA in their cell, it could lead to potentially some sort of generation of cancer or other safety concern. So this is very front of mind in many of the therapeutic instances, and people have been developing very great ways to predict and profile these off-target edits, is what they're called, as well as engineering the actual enzyme itself to increase the specificity and increase the potential safety of it. So it's really kind of a great broader question for any technology developed. You need to be able to profile how well it works and how that couples into both the effectiveness and safety of that, especially when it's a therapy, of course. And it's always something to remember that every technology is a technology in process, right? We used to have a Illumina MySeq in our lab, and now we have an XSeq, and these things are always scaling up. And so it's important for biotechnologists and other technology developers to always understand what the next demand is. And so CRISPR is like any other technology in that respect.
Oh, that's excellent. And so our next question is for Dr. Ashley Van Zeeland from Illumina. So what do you see as like the top 3 exciting advancements as coming from the advancements in sequencing technology?
Oh my gosh, it's hard to narrow it down just to 3, but I think you've heard from the panel today that the concept of now treating DNA, RNA, any omic in the body as a real kind of software system where you can read, write, and edit, and then go back and do it again. I think is hugely exciting. So you've heard about CRISPR applications. I think CRISPR is going to be a big deal, both on diagnostics and therapeutics for the foreseeable future. Cande talked a little bit about other forms of synthetic biology, right? So really thinking about how do you engineer circuits and functions, new therapeutics. I think biopharma and synthetic biology is going to be a really big deal. And also just, continuing to explore that relationship that we saw from Dr. Shirasu-Hiza around the central dogma, right? DNA is a critical part of this, but RNA is hugely important, right? I mean, look at what's happening today with mRNA-based vaccines, and then the proteome and how you go from what's in RNA to actually what's doing functional work in the cell. That whole space, I think, will be incredibly exciting. And so underpinning all of that, of course, is data science. So that's kind of the platform, I think, that will drive those three areas.
That is incredibly exciting. I remember when I first was in college, I was told 95% of our DNA was junk. So I hope that after this webinar, everyone understands that that is not true. So we have one last question, and we do have a lot of high school students and teachers joining us today. And the main question is, how do you get involved if you're interested and you may not have tons of access to everything? How does a student start out and become you? So we will start with Dr. Shirasu-Hiza for this.
That's a really great question. I was so lucky to do RSI, but there are many programs actually out there for allowing students to do summer research. And I would encourage people to look into those local programs. You can also do experiments at home. So I answered a question like this in the Q&A, right? Circadian rhythms is so accessible for kids, especially because everybody's fascinated with themselves. And it's a very easy way to get kids hooked into the idea of observation, hypothesis generation, come up with an experiment, and then try a simple set of variables to control for in that experiment, and then see what the result is. It's really simple to do. For example, my son, for a science project, charted he's not listening, so he won't be embarrassed, but he charted his poop. I had him make a poop clock, right? I said, when do you poop? Just write it down every day. Whenever you poop, if you poop, write it down. What time of day? And it turns out he has two pooping times, one in the morning and one in the late afternoon. And he was able to show that there was a bimodal distribution. And I mean, he did this for a month. So, and then, I said, well, you could start thinking about whether that's related to, for example, the actual time of day, or is it more related to like the last time you ate before you pooped? I don't know. So anyway, these are the kinds of things that you can get. You can just very simple, pooping or sleeping, time that you spend walking, or time that you spend on your phone, or time that you spend doing a particular activity of any kind, right? And then is there a rhythm to your behavior, and how is that rhythm affected by changes in other parts of your day? I think it's fun too.
Thank you so much. I do not want to track when I'm on my phone, because that's some piece of information you just don't need to know. So Omar, how did you get started? What would you have to suggest to students?
Yeah, I mean, I think similar to Mimi, I would say definitely in high school, look up as many research programs you can. And if you're also fortunate enough to live near a research university, I just emailed a ton of professors in the biology department at Western Michigan University, which is 20 minutes from my house, and ended up researching medicinal leeches and their neurodevelopment for 2 years, which is awesome. It wasn't just a summer program. I would go there every day after high school ended as well. And so that's how I got into science fair and ISEF and all that. It was just basically just going to my local university. And then I would say just a lot of labs like sometimes don't have room for like high school students or you know, people who don't have as much research experience. So like it's just more about like the numbers of like emailing like dozens of labs and like postdocs and grad students and just like not giving up. I mean, when I was a freshman at MIT, it was hard for me to get as a freshman into a research lab during the year. I think I had to email like 20 different professors and most of them didn't respond. So, and then like that summer I wanted to do research at NIH and I had to email like another like 20 labs. I would actually even cold call PIs because a lot of times the people wouldn't respond to emails. So, and then like once you know, get a bunch of research experiences, it becomes a lot easier and just, you know, pretty natural at that point. But that's, I guess, my advice. I don't know. Yeah. Try a lot of stuff too.
Yeah, just throw it all out there, see what sticks. I also had to talk to a bunch of professors to get into research. So persistence, yes, just because you got to know doesn't mean you're not good enough. Jonathan, do you want to just, let us know how you got to where you are and what really helped you as a high school student?
Yeah, I mean, obviously those points that are already brought up are super important. I think being able to kind of just do science projects at home. I remember one of my first science projects was I was actually toasting bread and looking at how jelly absorbed into it. It's actually a pretty complex process involving the Maillard reaction, but it was really easy to do and weigh, and it involves a lot of scientific concepts, and certainly being able to find research opportunities or other opportunities to do research or study. Great mentors are also important, as Omar said. I grew up a little bit near NIH in Bethesda, so I was able to go there. And also had a really great mentor at my high school who obviously allowed us to do things like USABO and other aspects there. But I think one additional aspect is both, you know, just as a social activity and as a great way to meet future collaborators. Science is, you know, a really collaborative process, and it's one of the most fun things about science is to interact not only with great mentors and be in great research opportunities, but also with your peers. And I mean, that's, you know, one of the great things about RSI was you can kind of make these long-lasting connections. But even in the absence of an environment like that, you can try to find people at your school who also are really into stuff. I remember when I was a senior, I actually kind of bound together a group of people into like some sort of biology club who would, you know, study for things and do experiments. And it's really great to learn how to work and think as a team, because science that's what it is. It's about collaborations. It's about sharing information. It's publishing and bringing that into the world and seeing what comes back. And starting that early and getting that really great feedback from people who love it just as much as you is really fun. So reach out and find other people your age or around that, and you could do that. And there are competitions that involve this. iGEM, which is the International Genetically Engineered I don't know, but it's a synthetic biology competition. It's really great. They traditionally do undergraduate, but they have high school level competitions now, so you can actually band together their tool and team and do something cool, like, you know, make E. coli smell like fruit. I know a group that's done that. So yeah, there's, you know, it's always fun to do something amazing with other cool people.
Oh, thank you so much. I think that's, you know, a wonderful theme is how collaborative science is and just meeting and networking with people. And if someone will make E. coli smell like fruit for the future, that's a worthwhile change because it does not smell good. So I want to thank all of our amazing speakers for joining us. It was so incredible to see how genomics and genetics is moving forward, what we're learning from it, and what we can accomplish in the future. And I also want to thank everyone else for joining us on this Sunday to celebrate DNA. I do want to say that this is recorded, so we will be sending a link to the recording to everyone who registered. Also, 5 lucky individuals will be winning a raffle for our DNA themed science art, which we will be contacting you with. And just as a final saying, we hope that what you take with you is the incredible amount that we have accomplished in the past 20 years in the world of genomics. But it has left us with so many questions. We need inquisitive, passionate, and creative individuals to help us answer and move forward. And this could be you. So please reach out to CE if you're interested in any of our programming. And we hope that you have a great rest of your Sunday. Goodbye.